on the list, but I'm talking about it besides that you can see the individual vascular bundles, of course. And then the bud, right, at that node.
Can I ask a quick question? Small nit on that, maybe I misunderstand, but where the leaf scar is, that's a small auxiliary bud right above it, right?
Hmm?
So then the inner node should be two different inner nodes, but
You're
not
right.
spanning that whole thing.
You're right, both this one and this one
Yeah.
and this one.
That threw me off too a little bit.
Yeah, sorry, this is, I grabbed this from another book, and you're right, that's not a great example. If you corrected it, absolutely, and if you answered based on, hey, that's the one left, that's pointing at the big thing, also, okay, but that's right. Inner node, we're talking about between two nodes only, so you got here, here, here. Anything else on that one? Definitely know those. They'll connect to the leaves as well, of course, because the leaf scar is there, right, and they're moving outward into this material here. So, roots, you're thinking water uptake. Leaves, you're thinking photosynthesis, absorbing sunlight to create plant food that then we break apart for energy. In the process of respiration also takes place in the leaf, and the products, the gaseous products, are released through the leaf in transpiration, as well as that water that's being pulled up through the plant. Always, I showed that pic from the book on 174 of the movement into the root and out of the top. That's transpirational pull of water. So the leaves, right, they've got incredibly important roles, and when it comes to all this, their positioning, their size has a relationship to what it's doing here, right? More surface area, more potential photosynthesis. Likewise, more transpiration, losing more water at the same time. So there's a connection between the design of the leaf and its function, and that varies from leaf to leaf within a single plant, as well as species to species, and certainly from climate or biome to other climate zone or biome. Yeah.
If I'm not correct, like the respiration and transpiration have to be like on a certain type of balance or something, or what is it?
There are
that
two
has-
separate processes. So transpiration is just that water pull, the evaporation of water
Yeah.
from the plant. And respiration is we say plants take in carbon dioxide and give off oxygen. It's the respiration is where they break down the photosynthesis products, the sugars that are made, into available energy. And in that process, they release that oxygen.
Well, I think I'm just talking about the transpiration rate.
Okay.
Isn't it something environmental where like the transpiration rate has to even out with the basically like if the plant's not if the plant is transpiring water faster than it's getting it, then it kind of makes the plant die.
Well, it definitely makes the plant wilt.
Yeah.
Yes, so these are two active processes from the plant. This one is absorbing energy, and this one is harvesting that energy for use. Transpiration is just the physics of the environment that the plant can't get away from, and that's going to be impacted by the relative humidity of the surrounding atmosphere, by the temperature, by whether it's shade or direct sun, and which proportion of each. And so that will impact how quickly that water is moving out. Yeah. And if it can't keep up, right, then there's not enough molecules in the xylem chain, and then the effect of those gaps, that cavitation, right, air bubble is going to be to start to wilt.
So yeah, it's like go either way, right? Vice versa, the same thing. If it's too much water and it can't transpire it quick enough.
Well, the thing is, if there's too much water, it can't transpire any faster than the environment allows, right? So it can't account for that internally.
Okay.
So then it can get waterlogged, right? And you see then some of the same symptoms that might show up if there was not enough water. So sometimes right off the bat, you can't immediately say, oh, that's overwatering or under, because one of the first things is wilting in both cases. Yeah. Let's get into it. knowing all the types of these, I'm gonna show you like, hey, we got words for all different kinds of leaf bases and leaf margins, etc. This is not just a plant ID class that's concerned with that. I just want to get to the point of, hey, there's different words we use for classification here, and they help us determine what thing is what, and there's a connection to function. So the basic makeup of a leaf includes the blade, right, that's the big part. The sheath in grasses, we talked about last week. I peeled off those big grasses and showed you the part that's still a leaf, but it looks like it's along the stem. That's the sheath, the midrib, right, the big vein in the middle, the petiole. Many have this, right? It's a connection to the shoot, to the stem, still considered part of the leaf. Stipules, sometimes they have these little guys, an ID feature for some families. Axillary bud, may be present. Bases, that's like this is the leaf base. Veins, different pattern of veins inside the leaf. The margin is the outside edge, and we have different words to describe what that looks like. And then the apex is the tip. So you're digging in deep on plant ID. How many are in plant ID right now or took it previously? So almost everybody. So these are the things where you might make a distinction between two plants in the same genus because the leaf margins look different. Or you might distinguish between families because the vein pattern is different even though the general leaf shape is similar. But here we're just introducing those facts and saying that we can use them in terms of ID, and there's a connection to the plant's function. So one big division here is leaf type. Is it sessile or petiolate? Petiolate leaves have a petiole. Right? Again, that's the connection between the leaf blade and the stem. Some do not have a petiole, and we call those sessile, right? Directly connected to the stem. That's all that is. Easy distinction to make in some cases. Is it petiolate or not? If it is, right, that rules out a group of species in terms of your ID. And it has other uses too. Of course, if something's further out from the stem, long petiole, maybe it's gonna fall down easier here in autumn. Maybe it's stretching out, it's useful, that's adding, it wants to be further from the stem for more light, so that might say something about the climate that it's in or the conditions. But one's directly connected, one has a petiole. The petiole is part of the leaf. Leaf types, you've got simple leaves, right? Or compound leaves, multiple leaflets. So 1, 2, 3, 4, 5, 6, 7 leaflets in this one leaf. One leaflet in this one leaf, which we call simple leaf. Some compound leaves are further divided into palmate or pinnately compound habit. This is pinnate, where there's I'll look like this, one opposite the other here, and palmate, like the name implies, that's like it. Everything comes from this central point, but there's still multiple leaflets connected to the stem via the petiole. So that leaf is compound and palmately compound. This is pinnately compound. Trifoliate leaf is just a specific case of pinnate compound growth habit with only 3 leaflets. And then it goes one step further, there's bipinnately compound, so not only does it divide here, right, like this, but then on that same piece, right, it also divides into more tiny leaflets. Just ID features to be aware of. The petioles here on these compound leaves. After the petiole is the rachis. Again, this is all ID specific. For us, we're just knowing that these are different types of leaves that are out there.
The rachis, is that that a secondary, the branching stem, it looks like it's pointing to a petiole.
Mhm.
So just further.
This is just the petioles from here down.
Got it.
Yep. Like this. Minor distinction, plant anatomy people, plant taxonomists need something to do. So in terms of veins, we've got parallel veins, we often see that in grass species, pinnate veins, right, again, that opposite division, and palmate veins, just like palmate compound have it, right? They're all coming from that central point, just like all of our fingers on our palm are coming from the hand, one way to think about it. So again, here, now we're gonna ask you, like, what is an arcuate venation of something? But just showing you, we got names for all these things, to get very specific about what we're seeing. But you know that we have sometimes parallel, pinnate or palmate, good enough. Different leaf veins, classified different ways. And related to that, the leaf ID homework is pick a sample, and record the following information. Either draw the leaf, add a photo, which you could print and staple on, you could upload in Moodle, etc. But I need something to look at to see if you've correctly identified the things. So that's within the individual leaf, right? Some are compound, some are simple, different pieces. Outside, we talk about these leaf arrangements. You know, about alternate here and then over here, node, and then over here, node, or opposite leaf arrangement at the same spot, opposite one another, just like the compound leaves from the last one, or world, right, where these are sessile, right on the stem, and they appear to be in a palmate fashion right at the node. You know, relatively few examples of world leaf arrangement. Most of the time when writing something, we're saying is it opposite or alternate. John?
How do I tell that it's the world versus compound?
Good question. Anybody else? How do you know a world leaf versus a pinnately compound leaf, or are you saying a palmately compound leaf?
The palmate is, and kind of combining that with the bottom picture you have there.
Okay. Thoughts?
Because of petioles.
Yeah. That's it. You got the petiole that distinguishes this whole thing is a leaf. And
The world doesn't have a petiole.
no, sessile leaves directly connected to the stem. So leaf, 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 leaf. And then for what we just looked at, leaflets, 5 leaflets with that petiole connection. So world, alternate, opposite, 3 different ways to see how the leaves are connected to the stem. Again, connection directly to survival strategy. Alternate, of course, you're making space, right, for light absorption. You've got an open area here, and then right, you're using different sides of the plant. Opposite, things are stacked up compared to alternate, so there's less space on one side, so we tend to see more internode elongation to make up for that when more light absorption is needed or under shadier conditions there. And in the world, there's no direct competition here, there's a space, and they're at that same spot again, so there is some overhead shading, but they're limited by development here. They're not going to extend out any further without the petiole. And leaf shapes, again, not going to test you on leaf shapes, but you can get a little bit familiar by answering the leaf ID homework questions. Right? Wide variety, you can see from something that's chordate in the upper left, right, down to something very narrow and linear. We've classified all these things so we can all talk about the same stuff. So useful to be aware of, at least for the purposes of this class, answering the questions on this leaf ID homework will be the biggest spot where you interact with doing anything surrounding Leaf ID getting into these, which is the spot where you can look directly at the slide. You can look directly at and this embodies for gardeners 53 through 5 has the same explanation here, has classifications here. So spots to go to that info that don't require you to memorize all these things, just get familiar with them. And that's good enough. Leaf shapes, leaf margins. Again, we're talking about what does the outside edge look like. Oak leaves, we know are lobed. You know, the white oak types have this smooth edge, smooth, low, right, rounded with these bigger sinuses in between. That's what we call that space. Other things are serrate, little cherry, right, little sawed leaves. Sometimes we say they're toothed. They've got teeth on them. Some are doubly toothed. Even each tooth has even smaller little teeth inside, right, all the way to palmate. If it's smooth margin, smooth edge, no teeth, no lobe, etc., we say it's entire. Just smooth edge, entire leaf. Leaf bases, again, just different ways to think about them. No need to write them all down or memorize these in here, but just to get the idea that there's differences among all of them. And you can refer to this as a homework question. The apex, again, the tip, same thing, from flat to very pointed, rounded to not so rounded. We got words for all this stuff. Questions on ID so far? Yeah, that's straightforward, right? Just look at it. You've heard it once in plant ID. And now let's take 10. And at 2, I'll start talking about the biology question. Oh, the old ones. Yeah, that's another reason why I wanted to get these, right? Because you can really see it
But it's
clearly.
like locust that, like, I think I put it on label so you can see the reaction
Yeah.
growth too. I'm gonna see if it's in there. Because we didn't remember. I never really got this way, the differences, which is cool because in the end, we found out how much they work together so similar. I learned, yeah, so you're just like, the same thing, stems, benefits, and then mature stems, and that's what our question was. And then you have to.
Right, absolutely.
So that's what I did. I didn't know if I wanted to take a picture of it yet, because we were in town. I might have wrote on the dock when we were going through things and just please disregard my scribble. But I just wanted to let you know I came through. Just put it over here. My brain doesn't work fast like that. My brain just doesn't work fast like that. I have to go home and sit on the couch and clear my brain. Okay, what am I actually reading right now?
I mean, having focus is important.
one seed gets you one little plant, you can't take that kind of time. Yeah?
Yeah, little girl.
Wow,
8
congrats.
pounds. Woo.
Okay.
You know what's funny? In my mind, it's gonna be this big in the classroom. It's giant, but it's cool. A black locust. But I just thought, we should just stash it in Jim's office.
Yeah.
But I think it's really cool seeing the difference of like where it was, how it was reacting.
Even this, right, there's, part of that is.
Isn't that so crazy? That's why I took this bet and I was like, this happens all the time where I'm in the field, I'm like.
So what was the situation that this side, wasn't growing and this side was growing fast?
The whole thing was sad.
Yeah, but there seems like there was something about the location specifically, right? But this side wasn't, hardly packing any.
I think that was the interior area and it was like growing out
Like
like
right
that.
up against, was there
Something else.
trunks maybe?
Yeah, there was a lot of stems.
Mhm.
This was a branch.
Uh-huh, okay.
So,
Branch.
yeah, but. But I'm not the most, like off the top of my head, I can't, yeah, I think this was the inside area, but
Yeah, something here was making it so it couldn't
You know how,
do
like,
that, right?
soft
Because you can
goods
see,
and
like, this is a good year, right? Lots of growth. Yeah, sorry, go ahead.
But like the softwoods and hardwoods, their like reaction growth is like opposite or whatever, or like different, so I'm like, I'm confused.
Mhm.
But I think it's really cool.
Yeah, I mean, this would be a hard one, right?
Yeah.
Not the hardest of hearts, but like, definitely a decision.
Hard enough,
Yeah.
but
It's really get to look up here, right?
it's like so cool how big I was even looking at costume.
You can see here, this is what I was saying in here, it's much more visible in this one. You can see the early vessels, much bigger, and the traces, like the little lines.
Cause I cut some, cause I was like, oh, I was thinking about getting it. And like, still like those, shiny. And
tree, there's a little bit more xylem that's functional, maybe not as much, but, I mean, it's still like, right, it's not like 6 inches or anything.
Science maybe, yeah.
See how it shows up.
What's cool too is that the center of that tree, it's like you grew out in one direction, huh?
Yeah, they use that,
Yes.
yeah, that's weird.
So that's a limb?
Something was in the way on this whole side where it
Yeah.
couldn't push,
Yeah, that way.
and we said there was several main shoots, so it was probably just like up against the other one. Cool, so we're back. Before we continue with leaves, Emily brought in this cool tree cookie off Black Locust that she cut. So that's Bovinian, Pseudoacacia, another hardwood angiosperm species. Really cool opportunity. I'll do it up here, then I'll put it on the table for everybody to see. So I guess first off, this thing's large, right? Several dozen years old here. And unique aspects. This is the original first year, right? And then some of these things, right, it's almost 3/4 of an inch really wide growth rings out here. But within that same ring, here's a really big one, and if you follow that over here, there's almost no growth taking place on this side. So something on this whole side of the tree, it sounds like it may have been another trunk, another shoot blocking it. Could have been a neighboring tree, right? It might be up against a house, right? Different situations will produce that. But even within the same year, very good growth year, couldn't do it here, so it was depending on the opposite side for producing that xylem, right, to move all that water and get the growth. Really nice example likewise.
You said this is several thousand years old?
No, it's
Oh.
several dozen years old.
I'm not about to say that, I'm not seeing the ring. Okay, count it.
Looks like maybe 55, 60.
Yeah.
Well, this was a branch up.
This is a break, okay, so
Up into the.
main trunk, yeah, could be well.
So if it was a branch, would it just be up and next, like another branch?
Yeah, I'm guessing that's what was happening.
So would that almost be like, like included wood at that point? Yep.
So hold on, let me ask another question. So if I chop a branch down the tree, the rings on that branch is how old the branch is, not how old the tree is.
That's right. That thing can't record history before it was born.
Yeah.
What does tropism look like with a branch? Would it look similar with the very little growing on one side, like if it's going up at an angle or something, or?
Like the growth toward the light.
Yeah.
It depends on
Depends on the
the
cross
same
section.
limiting functions, like was there something on one side blocking it? Otherwise, that is like a daily adjustment, so you're not gonna see, in the stored record as much of a difference there. But a cool opportunity to see the bark too. Emily correctly mentioned that my cookies from last week didn't have bark, so you get a nice look at that division, right? Just a little bit of living phloem to the lower side up there, and then the cork cambium, that additional meristematic layer that trees have, other plants do not, producing that bark to the top.
This is the cork barrier,
Mm-hmm. Like where it changes from light to dark right there.
right? That's the
Yep.
cork barrier.
That's the cork cambium, yeah. So really cool. I'm gonna move it back over here so everyone can take a peek as you go. You'll notice too, that very interior, it's cracked. That's common with the oldest stuff. It's dead now, non-functional. And it seems like like probably this thing was at an angle where they're both not growing there, and there was something happening that was giving some pressure that created the damage. So leaf biology, we're not gonna get into the function now of the things that we've named. And just like with stems, just like with roots, there's some differences between monocot and dicot. Here primarily, it's the presence of the leaf sheath, whereas in the dicot species, we don't see that same situation. Because there's more branching. More lateral branching prevents that need for the wrapping on that single stem of the grass species. Otherwise, all the terms are basically the same there on the outside. So let's go inside. And inside, many of these things, monocot and dicot, look the same. So let's get a general understanding of what's going on inside. Okay, first off, tissue types. It has epidermis, which was root hairs in the root, in the stem, and it's just that outside covering. Here, it's generally waxy. We call it the cuticle. And here there's kind of there's a cell layer, and then there's the waxy deposition on the outside of it. That wax is meant to keep water in, because these are the things that are exposed to light, the atmosphere, that are doing all of that transpiring of water out. So if the rest of the leaf can hold water in, that's beneficial, because that surface area is costly. Compared to the root, we don't have a waxy outside covering because we're not concerned about spending energy on something to preserve water loss when the goal there is get water in. Julian?
You can also add like an area or like a little follicle outside, right? Instead of the waxy layer, like doing the same thing.
Or a combination. Yeah, I'm getting there. That's coming up. So inside, we've got epidermis, then we've got mesophyll, which is the ground tissue, and here it's primarily the photosynthetic area of the plant. That's why it's specifically colored green. It has the green dots here that are indicating heavy concentration of chloroplasts that are doing the light absorption. And then notice this would be the top of the leaf, assuming it's somewhere out like this, waxy cover. The bottom has stomata. Some leaves have them on the top. Generally, most leaves have 80 to 90% on the bottom, because they're not in direct sunlight. They're not exposed directly to the atmosphere. So again, it's a way to save water, which is an important aspect of leaves that I'm going to ask you about in the homework, the function part, and address in more of the slides. So stomata are holes to the atmosphere. So that's free air passage. This area are all in direct contact with the atmosphere when the stomata are open. And so that's when transpiration is happening. That's when the release of the oxygen from respiration is happening. Of course, so that's epidermal or dermal, right, ground tissue. We still have our vascular tissue. Phloem on the outside, xylem on the inside, connected throughout the leaf. All those veins, right, the different vein patterns that had the names I showed earlier, serve the function of getting water to all these cells in the xylem and getting the photosynthetic products from the mesophyll into the phloem and moved elsewhere.
So respiration and transpiration would take place both through that stomata.
That's right. So you'll see the respiration, the production of the gases and the getting energy takes place inside cells, but the release of it is via the stomata.
And so that was my question. The respiration happens in every living cell, the trunk, the whole thing. But the release of gases, we're saying, is only taking place in the leaves, or?
Primarily, let's say, in the leaves. Some plants, and especially trees, or woody perennial plants, have lenticels, which is another like gas exchange hole in their stems or trunks. But On leaves, it all happens in stomata for sure. And the majority of plants are doing it almost exclusively through stomata. And yes, so respiration, well, photosynthesis is specific to plants and a few photosynthetic algae. And respiration is all organisms, right, that are using energy. So we're respiring to get energy just like plants are. And that's why we all have those powerhouse organelles make energy. The mitochondria, that's right. So again, different ways to look at it. This one again is actual cell stain. Not my favorite, but I think between the others, you can get it, or maybe this one works for you. Seeing the internal structure. So I'm comparing monocot and dicot here. You can see in the monocot, we've got vascular bundle, phloem on the outside, xylem on the inside. They work right as a unit, this tube moving around throughout the leaves. We've got the mesophyll, also here that's called parenchyma, divided into two groups, the palisade and the spongy. And then the stomata, they're surrounded by a guard cell. The stomata is the hole, right? You can't actually point to anything. It's the opening, and the guard cell is what controls if that is open or closed. And I'll talk about that function too. In the monocot, same thing. They bundle their vascular tissue, and there's just a little bit less space. Most of these leaves are thinner, but relatively little difference compared to what we saw in stems. Take a specific look at the mesophyll because it's an important function in photosynthesis. So again, two groups, the palisade, right, long and skinny, up by the epidermis, up by the surface of the leaf, and then the spongy, they tend to be round, they tend to be looser, packed in the more open area on the bottom side of the leaf. And both of those things contain the chloroplasts that are the organelles absorbing the sunlight and producing sugars for energy from that. So these cells, like ground tissue elsewhere in the root, in the stem, can be used for food and water storage. So again, that connection, similar function among the ground tissues, different name, different place, but stacking of that same type of function. So here, two types of ground tissue, both photosynthetic. Which do you think, spongy or palisade, most of the time, does more photosynthesis? Palisade, and why?
Closer to the surface.
Closer to the surface, exactly, right? That's where the light is available to absorb. So in some cases, right, some makes it down here and it gets absorbed. In some cases, you might want to move produced food over into these because they're then connected to the vascular system, right? The flow of that can transport them. The palisade mesophyll, by and large, are doing the majority of photosynthesis because of their location. That means they have the majority of the chloroplasts, and why their structure is skinny and long, so that they can be catching all that light as it penetrates through the leaf, right? And then whatever's left over here is likely not as energy packed, and so fewer chloroplasts needed in the spongy mesophyll to do that. Here you can see better the stoma 1, or stomata plural, is the opening in the guard cells inside epidermis control the opening or closing. And I got a specific one for it. How that works. Right up. Question? So this is just what I was talking about. Most of the photosynthesis in those palisade cells. Again, just another opportunity to look. So here's a leaf that has stomata on both sides. Mesophyll tends to be compact, right? Some are elongated and lined up close to each other. The others can be stacked on on top of each other. Tend to be more loosely packed down below, tighter packed up here. And if for some reason the light source were to change, let's say this is a house plant, a greenhouse plant, something where, for whatever reason, that artificial light source is in a specific location all the time, some of those leaves are both working toward the light, but conditions might be such that they're always, the underside is facing that lamp, or it's facing the light source, then you will start to see these chloroplasts migrate, so there's more in the location that has the direct light exposure. So again, these generalizations about the cell roles, which ones are doing what and how much of it, are based on natural growing setting outside with sun as the light source. And once you start limiting them inside, it can adjust. I mean, same thing outside, right? If we're in a shade situation, it will adjust. But we're talking about the basic way that the plant is set up to thrive based on this arrangement. So when it's leaves, we're focused on photosynthesis, getting that sunlight, number one, making that food that then we can get energy from to run the plant, to grow, to put on, to repair damage, to prepare for the future, etc. But don't forget, all of that space has to be reached by the vascular system. And so in leaves, we see the xylem phloem encased in what we call the bundle sheath, which is just like the circular layer on the outside that keeps it all as a solid unit of vascular tissue on the inside. It runs throughout the mesophyll to contact all those cells, because that's where the food is transported, that's where the water is delivered. And so contact with those cells, it has to be at least, minimum a few cells away so that it can carry out those functions. If you get too far, it's just like blood, right, in our own body. If cells are too far from the capillaries delivering oxygenated blood, they're going to suffer. And it's going to be extra energy to move oxygen cell to cell to cell instead of having a direct connection with the capillary or with the xylem flow. And
Just maybe an obvious question, but we're saying that every single vein in a leaf is composed of both xylem and phloem. So-
yes, yep. Because why are they together?
They need every cell needs water and nutrients.
Functionally need both of those all the time. That's right. And why else?
Because once the chlorophyll, once photosynthesis happens, that energy will be transferred through the phloem and to other parts.
That's
of
what
the
happens.
plant.
What I'm getting at there is because that cambium layer is in between them, and that's the meristematic tissue that makes them both, right? They always appear together because it's one cell layer making xylem, one cell layer making phloem, and so if we extract that and you split the meristem, it's not going to be functional for both anymore. So it always appears as this unit, and in the leaves, where we're more exposed to water loss, especially from the atmosphere and direct sunlight, then they're bundled together to keep that water from escaping as much as possible. So another view here to show you kind of the space and talk about the stomata in particular. Right? So an important function of leaves, these openings that are majority on the underside of the leaf, bottom of the leaf, away from the direct sunlight, right? The same reason that chloroplasts are in the palisade mesophyll on the top, toward the sunlight, but here, reduce direct sun contact and reduce water loss. Exchanges gases, right? Talking about oxygen moving out, of course, that's also how carbon dioxide moves into the plant, that goes into photosynthesis. That's the start there of getting that carbon molecule that's gonna become the sugars, gotta get it in, and the stomata is the place where it enters. A single leaf, of average size on a tree out there has millions of stomata. So lots and lots of these undergoing the same process at one time. And they don't all change. It's not like an on-off switch where every stoma on the leaf is either open or closed. It's a relative amount. So when it's very dry and the plant is losing a lot of water, it wants to close stomata to avoid losing some of that water. But it can't completely shut them all down at once because then there's no carbon dioxide getting in. So it's like a relative, maybe 3/4 are closed and 1/4 are still open, so it's reduced that loss. But it's not an all or nothing question because the quantity is so high. Lots of these on every leaf, most on the bottom. They're controlled by the guard cells on the outside that form the hole in the middle. And sometimes these stomata are sunken, which is like recessed into the epidermis as another way to conserve water. Claire, any Christopher?
So you were saying that there's no occasion where all of the stomata are closed.
I mean, there could be, but in general, it's a fluctuating relative amount.
So like in a vineyard, once it gets 110 degrees, we say that everything is shut down in the plants. Is that an instance where all of the stomata are closed because the plant is just trying to conserve water?
Yes, extreme heat would be a good example. And of course then, so yeah, it's not at all to say that that can't happen. Say under most circumstances, you still want some carbon dioxide moving in, but extreme heat like that, especially if the relative humidity outside is quite low, which is like in our summer when you're getting that temperature, that's what it's like, you would be losing water really quickly, and so shutting them all down is an effective way to stop that. The downside is now no carbon dioxide in means no new energy can be produced from the sunlight that's getting absorbed, right? Once whatever has already gotten in gets used up, no more, right? No more fuel for that fire, at least until summer reopens. So that's when everything is shutting down. Christopher and then Ryan.
Trying to understand the gas extinction, was wondering if the spongy mesophile being so close to the stomata, does that mean that's more responsible than the palisade mesophile for the exchanging gas?
Yeah, that's right. If you want to get something out, much easier to do it here. You can dump it right into this open space directly. And likewise, also more risky to be closer to the opening, because then when that pull of high temperature and low humidity is there, it's pulled directly.
So like you said, you gave the example of like turning the plant upside down and the mesophile tissues would migrate and try and right themselves. Would that be why, or is that just because they're trying to get light?
Yeah, so what I was saying was that they'll move the chloroplasts into one or the other, wherever the light's more direct. But they probably won't fully like rearrange, they'll just shuffle the chloroplast concentration. Right?
So during winter with the angiosperms, when they drop all their leaves, does the respiration then like turn into the cork? The respiration?
Like where does it happen now? Yeah. Then in these trees that we've been talking about, then it moves to the lenticels in the trunk. Yep, in those openings in the could be a woody branch, the main trunk, wherever. But great question to bring that up. And this is the difference then between the gymnosperms and the angiosperms here. Is gymnosperm strategy still has all its needles, right? it's still gonna be absorbing some light whenever it's there. It's gonna be like, you know, lower angle, less energy light that it's getting, but can do a little bit of growth slowly. Meanwhile, the deciduous angiosperm is effectively shutting down. Now, metabolism is not zero because it would be dead, right? But, you know, much, much slower than when it had a leaf canopy and some sunlight for active growth. And so now, all it's doing is respiring, so it needs some gas exchange opportunity to let the oxygen out from the slow metabolism of that leafless tree that's going into wintertime, but it doesn't need to get any carbon dioxide in because it has no leaves to get sunlight to make that energy with. So it needs a lot fewer opportunities to let gas out without any leaves, and thus, you know, we call it dormant. It doesn't mean that there's no activity, no metabolism happening, but it is like, you know, 5% to 10% of what it would be during active growth. It gets higher, exactly. I mean, I think dormant is an appropriate term. And that really highlights the strategic difference in the approach to growth between the gymnosperm and the angiosperm there. Among, right, the woody trees now, we're talking about slow and steady versus fast and, you know, lots at one time. So, I'm gonna talk about the stomata and make sure I describe what this control is, what it means to be sunken, and remember that all of this is surrounding control of water loss. Huge for the plant. We spent that time at the beginning, talked about the importance of getting the water in, the importance of maintaining that xylem chain of water molecules, and now we want to use all that work, right? Manipulating the gradient, get it in the root hairs, moving it past the Casparian strip into the xylem. We want to make use of all of that, or as high a percentage as we can, without just directly letting it evaporate into the atmosphere through transpiration. So all these little methods to hang on to more water come into play to increase the opportunity to do more growth, right? And to reduce the need for water then from the roots, because we're still using some internally in the rest of the structure, the stem and leaves of the plant. And so I'm going to talk about some methods to do that. There's a homework question that addresses that same thing, and the stomata are the place where that water is lost. So we focus on how to control those and what forces that change. And here's a cartoony look at them. Remember, stomata is the opening. The guard cell is two plant cells, one on either side. When the stomata is open, they look like big C's. When the stomata are closed, they're slumped together, so air can't pass. So you can see in here, right, the big thing is the vacuole, and that is what controls the stomatal opening and closing. So when stomata are open, when stomata are open, water can escape, right? When stomata are closed, water can't leave, carbon dioxide can't enter. And so, in these guard cells, that vacuole, again, misrepresented here as smaller than it is, really like 90% of the internal structure of the cell. When that vacuole is turgid, right, aka filled with water, it's pressing on the cell membrane fill that space and make it rigid enough to be open. When there's not enough water, and we lose water, end up here. Now the vacuole is probably what's being shown, smaller, and thus that loss of turgidity, that loss of pressing like a balloon on the outside of the membrane, holding it all, right? Open, now slumped together. It's like they lost, right? The balloon deflated and hit another deflated balloon that are touching, and now, right, air can't pass. Does that make sense? So I think for some, it maybe feels counterintuitive that it's when it's filled with water is when it's open. So just like when other plant cells are turgid, they're upright, right? It looks strong and full, and when they don't have enough water, it looks wilted. You know, it looks slumped, crinkly, right? Same thing here, not enough water. That stomata closes. Yeah.
So does the pressure of the water kind of like, almost like push out from it?
Yes, it's literally pushing on the cell membrane, right? Which is then pushing on the cell wall, which can't be, if you push too much on the cell wall, you'll break it, you know? But the membrane is flexible. And so that's flexing to be as rigid and open as it can get.
Is it kind of like one of those like spray bottles that like when you're applying pressure, it doesn't want to spray, and then like once you apply, it's like the sports water bottles. Like once that pressure's been passed, it starts spraying until like the pressure has been equalized and then it seals back up. Is that like kind of the same thing that's happening?
Yeah.
Okay.
You think about another analogy for that? When it's filled, right, it's rigid and open, and when it's not, then it's slumped shut. If you think one, let us know. And your new analogy is useful. Yes. Question we hear in the back was, is the vacuole using salts to control this? And it absolutely is. So here, talk about salt concentration. We're also talking about just the raw quantity of available water. So, of course, under dry conditions, right, the problems that the plant encounters really start to stack up. Now, you gave the example, Claire, of the 110-degree day, and right, all these stomata on the grape leaves are shutting down, closing. So, the ground is getting drier from hot weather, right, and likely little rain. The atmosphere has low relative humidity, so it's pulling a lot of water. So running out of water to then fill these things and make them turgid, which means they close. So then we can't get carbon dioxide in to make more food. So it's both growth is slowing and new opportunity for growth by not getting that CO2 in is slowing. So things start to stack up as like this plant is slowing down overall now.
So it's because transpiration is happening because you have gas and water coming out of the top of the plant. Like that's why you're not supposed to water during a heat wave, is because the plant's not gonna be able to take it up.
Yes. So there's a couple reasons. Biologically, that's the reason. If stomata are closed, or if 3/4 of stomata are closed, well, I mean, the water is still coming in through the roots, right? But the CO2 that combines with the water and the sunlight to make sugar in photosynthesis is going to be limited there. So it can't take full advantage. So things are slowing down. And then the other one, and I think this is over stated and less of a risk, is that you'll get that magnifying glass effect of water droplet burning plant leaves, which I've seen very few times happen, but, of course possible. So to your other question though, when we need to get a lot of stomata open, we need to get a lot of carbon dioxide in to do a lot of growth, then we might need to manipulate that gradient and say, just like in the root hairs, right, lots of salts here, so water, you better go dilute that, move into the guard cell vacuole, increase the turgidity, and open that stoma. So that's just like with the root hair cells getting water in. There it's for just the plant absorbing the water in the first place. Here is to use that water specifically to open stomata. Julian?
So, does that mean like this is kind of happening naturally per stoma, or like is there more of a conscious thing happening with the plant and detect the lack of whatever is happening when it needs to open and close? And then tell us
The
if
plant is signaling all these guard cells to do this.
It's not just like a localized thing happening.
It's not like a physical response to the environment, it's a plant moderated opening or closing. And it's was affected by what the temperature is, what the humidity is, are we in direct light or not, all those factors. But in the way that transpiration is just happening, because that's the difference in pressure between the atmosphere and the plant, can't get around it, that's not the case here. The plant is saying, close them up, open them up, do a little of each. So again, these are guard cells. Just like the Casparian strip is guarding, sort of bouncing, right, the water entry into the xylem. Here, they're guarding the passage of gases, including water vapor, in and out of the leaf. So really important. I'm sure you understand both why they might want to use this, what it means to be a turgid cell, and how that when it has enough water, then it's open, and when it doesn't, that's when you get closed stomata. Yeah. Talking about all this, and I should have jumped to this one. So Julian, this gets to your question. Light is a dominant signal to open, so it's in response to, hey, getting a lot of direct sunlight here, so I need to either shut and hold on to water, or I need to open up, because that means, hey, if there's a lot of light, this is a good opportunity for growth, so I want to get some CO2 in and get this working. Solutes accumulate in the guard cells, which means osmosis moves water in, builds that turgor pressure, and the stomata open. Here's just an example. Don't get caught in the details, or I'm not going to test you on this graph or anything, but just to show you by time of day, so this is 1 p.m., 3 p.m., etc. More stomatal opening midday when growth potential is the highest, more closing at night when we don't need as much gas exchange, we don't need as much CO2, because we can only do one step of photosynthesis in the dark. Just to say, hey, right, it's no accident, no coincidence that it looks this way. The plant has specific needs, and it moderates that need by opening and closing all these stomata to get, or to stop water loss, or to get that CO2. Likewise, into your earlier comment, and this gets to our strategies by the leaf for water retention. We've got closing stomata now, making it so that we're transpiring less because it can't get out. Another is to make a little environment, a micro environment around the leaf that is more humid, warmer, less susceptible to change from wind, just a localized mini environment. And leaf hairs are one way to do that. Right? Different styles, right? Lots of small fuzzy ones, fewer bigger ones on the midrib, on the back side of the leaf. You weird trichromes, right, this harvest season. Hopefully you're using your 30x Hamlin or more to evaluate those things. Think leaf hairs, that's what they are. Right? They're an extension of epidermal tissue, just like root hairs are epidermal tissue. And that means they come from the epidermis. And they're making this little microenvironment, lots of little hairs, where moisture might get caught in there, a little more humid, a little less water loss in that local, right, very local, small area, but that can make the difference between needing to open stomata or keeping them closed. That can make the difference between, if you do this over the surface of all your leaves, a lot of water saved versus some water loss. Of course, you're reducing your photosynthetic capacity a little bit here. You've got reflective white hairs now on top of the green absorbing surface of the leaf, but because of the importance of water management, generally it's worth it.
So like for a hazelnut where it's got like fuzzy on the underside, is that,
It's kind of like this, but smaller.
so they're trying to reduce or manipulate the transpiration with those like.
Yeah, and not every leaf hair is exclusively that purpose. Sometimes it's to make it harder for an insect pest to crawl, right? Like little spikes.
Like a tomato and stuff.
Yep.
It doesn't work very well though.
Yeah. So sometimes it's a defense mechanism, but it always has the effect of increasing the localized humidity, and that can save some water while you're doing it. So reducing transpiration, aka saving water with the leaf hairs.
And then it creates that humid environment how again?
I'll show you. So, cloudy night versus clear night. Which one's colder? Clear night, losing more heat fast, right? Clouds keeping some of that temperature in. Clouds also keeping some of that moisture in. So it's like that. It's like holding it down. So if you can imagine, let's switch.
This is like almost creates a shade forest.
Yes, think about like a micro forest. I'm gonna draw a micro forest. So you've got the surface of that leaf, top or bottom, right? The effect is the same. So here's a petiole. It's gonna be a petiolate leaf. Right? It's entire. It's got typical opposite vein situation, but it also has all these leaf hairs. Okay, so now let's zoom in on a little chunk. Get the surface of the leaf. What's that?
I said, is it smooth or straight?
This is entire, smooth. So these are stomata in the example, and blue is the leaf hairs. Okay. And worth noting too, the difference in effect under the same structure, the same set of circumstances on the bottom of the leaf, and how that changes the efficacy, how useful this adaptation is. So, right, so these are, right, stomata are the holes, right, direct contact with the atmosphere. And now, in between here, I guess I've got to make my gases or water red. So as water is coming out, right, vapor droplet could get plugged up in here. In the same way that temperature change or humidity change forest, right, it's gonna be higher humidity under the tree than in the exposed area, 20 feet over where in the open field. So it's just little bits of moisture held in all those spots. But a million times over on that leaf, right, at this little scale, means lots of individual water molecules that are maintained that reduces the overall loss in this like little bubble, right? That could be, the quarter-inch of atmosphere around the leaf surface. But if that quarter-inch is the humidity level here is 80%, and out here it's 60%, that might be a lot of water that's saved here, right, and not directly pulled out. Does that help?
Yeah.
Andrew?
I was wondering, so I mean, with the whole leaf itself being part of the water absorption process, does that mean that these hairs are technically flowing water both directions then? 'Cause like as water sits on the hairs, does it go back into the leaf that direction, or is it only pretty much going out the leaf and protecting the water, too much water from escaping?
Good question. In this case, this water is not being absorbed by the leaf hairs. This is just a structure to maintain a humid environment around it. So it might run down, and be on the surface of the leaf, which would continue to keep the humidity higher, right, in this immediate area. But it's not then like transporting it into the leaf the way that a root hair is moving water into the root.
So it's not feeding the plant.
That's right.
So since it's epidermis, it would have holes in the cell walls to like let that penetrate back in.
Say again?
Since the hairs are like epidermis and that's how they're built, that's probably part of why it's like a barrier. It's not designed to be like a toothpaste or
Exactly.
or door at all.
Yeah, that's right. It's still part of that layer that is the divider between internal plant structure and outside environment.
Are there exceptions to that that are interesting to know?
Like plants that-
where they would reabsorb, like Andrew was saying, or is it just pretty much the physiology?
Well, no. Because you would need a root hair to do that, right? Yeah, so in this case, it does point out an interesting distinction because root hairs and epidermal leaf hairs are both dermal tissue. However, generally, this guy is exposed to the environment. We've got direct sunlight, we've got potentially wind. This is a big factor in drying things out, right? Wind, right? It's moving away moisture, it's cooling things off, which sometimes you want that, but it's definitely lowering the humidity in the immediate context. And so if you can keep it high right here, then maybe that wind is bypassing it, or it's not having as much effect further away. So my point is though, it's exposed to the outside environment in a way that root hairs generally are not, because they're inside the soil, right? They're protected from direct sunlight, they're protected from wide temperature swing, they're protected from wind exposure by being in the ground. So there's a totally different set of conditions that they're responding to, and thus the root hairs do move water in and then through to the next cells, right, as it gets through the ground tissue into the xylem. Here, that's not the case. It's just like an external structure that has this miniature forest effect. John?
So looking at like plant selection and evolution, would you expect that the plants that exist that have these kind of dramatic leaf hairs and one down in one, would you expect those to be more resistant to mildew issues?
More resistant to mildew?
Right, it seems like mildew would grow a lot more easily in that environment.
Oh, yeah. Yeah, no, I don't think they're necessarily more disease resistant, especially to molds or mildews. In fact, I think you're right? It's a calculated risk, because not only could spores of a fungus get caught in those same hairs and and start to establish, and then they would be next to a stoma, which would be an easy spot to get in. You won't even have to poke through any plant tissue, right? You just go through the open hole. So there is a potential downside there, absolutely. However, generally, I suspect when I see a plant that's so coated in hairs that it's a high sunlight environment to begin with, not the best place for a mold to develop in the first place. So likely that's not a problem. However, there's no question, right? This is going to reduce the wind effect and it's going to increase moisture, which is exactly what microorganisms are looking for establishment. So it's totally possible. But secondary to the importance of getting enough water, maintaining enough water, and then if you don't have enough water, you don't have enough energy to fight an infection later. So it's like, kind of take care of the bare necessities and then approach that bridge when you come to it. Other question about that? I think we're at time, so I won't jump into the last three. And that said, if you feel like you can't answer a homework question without those three, which are specialized leaves, shade versus sun leaves. I do ask you about that in the homework. Then I also ask you about racks. Try to answer it from the info in these slides here. Otherwise, if that's not working, answer the rest of the questions, bring it to class. I think there'll be enough info there to fill that last question in class.